The overall goal of this experiment is to study the activity-dependent nucleocytoplasmic shuttling of proteins using acute hippocampal slices where neuronal connectivity and function are well preserved. To achieve this goal, the hippocampus of an adult male rat is first isolated and acute transverse slices are prepared. As a second step, the hippocampal slices are transferred to the recording chamber, and the late form of LTP is induced in the CA1 stratum radiatum. Next, the potentiated and control slices are snap-frozen, and the stimulated CA1 regions are dissected in order to isolate the nuclear enriched fraction for further immunoblot analysis. Results based on Western blotting analysis show differences in nuclear phosphoprotein levels between the potentiated and control slices 30 minutes after the induction of LTP.
Generally, individuals new to this method will struggle because of two reasons. First, the low amount of tissue sample, and second, the critical time frame that is required for lysis of the samples in hypotonic lysis buffer, allowing release of the nuclei.
Visual demonstration of this method is critical as the hippocampal slice preparation and isolation of the C1 region require fast and very accurate dissections. But there are certain tricks to make it easier. One would need to follow both written and visualized instructions for preparation of the nuclear enriched fraction from the C1 area of the hippocampal slices.
Demonstration of procedure will be performed by Pingan, postdoc from our laboratory. She will show how to prepare acute hippocampal slices and induce LTP. After dissection of C1 region, Sujoy Bera, grad student from our lab, will show you how to isolate nuclear enriched fraction.